Billions set out on a microscopic odyssey. Only one will begin a new human life. In the next 7 minutes, we'll follow that single journey step by step, with real medical science explaining every push, turn, and signal. First, let's set the scene with anatomy and timing. Fertilization normally happens in a specific part of the fallopian tube called the ampullary region. This event has a very narrow window, just 12 to 24 hours after ovulation. Ovulation itself is when a mature egg, or oocyte, is released from the ovary, surrounded by a cloud of protective cumulus cells. After ejaculation, around 200 to 300 million sperm are deposited in the vagina. It's a hostile environment. Harsh acidity and the female immune system eliminate most of them almost immediately. However, during ovulation, the cervical mucus becomes watery and thin, forming tiny channels. These channels allow a small fraction, just a few thousand, not millions, to pass through the cervix and enter the safety of the uterus. Once inside the female reproductive tract, a crucial step begins: capacitation. This biochemical transformation takes several hours, typically 4 to 6 in humans. The sperm's outer membrane changes, shedding cholesterol. Its internal signaling pathways are rewired, and it becomes hyper-responsive, gaining the ability to bind to the egg's protective layer and undergo the final reaction needed for fertilization. So, how do the surviving sperm actually find the egg in the vastness of the fallopian tube? It's not just luck. They use 3 sophisticated guidance systems: rheotaxis, which is sensing fluid flow, thermotaxis, detecting temperature gradients, and chemotaxis, following chemical signals. Let's look at each one. First, rheotaxis. There's a very slow but steady fluid current flowing from the ovary down toward the uterus. Remarkably, sperm can sense this flow and are hardwired to swim upstream against it. This behavior is a major long-range guidance mechanism, ensuring they are consistently biased toward the fallopian tube where the egg might be waiting. Lab experiments in microfluidic channels confirm that sperm consistently orient themselves against the current. Next is thermotaxis. Sperm are surprisingly sensitive thermometers, able to detect tiny differences in temperature. Studies show that the area around the fertilization site is about 0.2 to 0.5 degrees Celsius warmer than the rest of the tube. Sperm actively swim toward this warmer zone. This provides another long-range cue, guiding them closer to their target, especially when a female is ovulating. This temperature sensing is thought to work by influencing ion channels in the sperm, which in turn alters the beat of its tail. Finally, chemotaxis provides the short-range, highly specific final guidance. As sperm get closer, the egg and its surrounding cumulus cells release a cloud of chemicals, most notably the hormone progesterone. This creates a concentration gradient, like a breadcrumb trail. Human sperm have special channels called CatSper, which are designed to detect progesterone. When progesterone binds, these channels fly open, causing an influx of calcium into the sperm. This flood of calcium triggers a dramatic change in behavior called hyperactivation. The sperm's tail stops its regular swimming motion and begins a powerful, whip-like beat. This high-amplitude, asymmetric movement gives the sperm a surge of force. It helps it detach from the tube's inner walls and, more importantly, gives it the power to push through the viscous, gel-like matrix of the cumulus cells surrounding the egg. This is a critical transition. A capacitated, chemoattracted sperm becomes hyperactivated just moments before reaching the egg itself. Once a sperm successfully penetrates the cumulus layer, it faces the final barrier, a tough, protective shell called the zona pellucida. Now, the acrosome reaction begins. Upon binding to the zona, the very tip of the sperm's head, the acrosome, fuses with its own membrane. This releases a cocktail of digestive enzymes that locally break down a small path through the zona pellucida, allowing a single sperm to finally burrow through and fuse with the egg's membrane. The moment one sperm fuses, the egg instantly initiates a lockdown. A wave of calcium ripples across the egg's surface, triggering the release of cortical granules from just beneath its membrane. These granules rapidly alter the structure of the zona pellucida, hardening it. This cortical reaction is the block to polyspermy. It prevents any other sperm from getting in, ensuring that only one set of paternal DNA contributes to the future embryo. After fusion, the genetic material from the sperm and the egg form two separate pronuclei, which then migrate toward the center of the cell. Their DNA decondenses, and they merge. The first mitotic division occurs about 24 to 30 hours later. That single cell, the zygote, has been formed, and it immediately begins to divide, starting the incredible journey of development. From billions, one succeeded, guided by flow, heat, and chemistry. If you found this deep dive helpful, please like and subscribe. And let us know in the comments if you